Welcome everybody, this is lesson 11 in our muscular system unit. We are closing down on the end of this, so we are back in the muscle and we are looking at how does this muscle actually contract. Last time we talked about the actual sliding filaments and how the actin and myosin are sliding past one another to make sure that the contraction actually occurs. So question is, how is this actually triggered? And the key to all of it is calcium. Now, when we mention calcium to most people, when you're talking about anatomy, most people think immediately of the bones. And of course, the skeletal system has a ton of calcium. But we also have dissolved calcium that is in our blood. And we have calcium ions that are in our cells as well. And it plays an important, very critical way of messaging inside cells, and particularly in muscle cells. So we'll talk about exactly how that happens. And by the way, just as an aside, when you are running out of calcium, of course, you actually replace it with some of the calcium that is stored in your bones. So you actually will, if you don't continue to supply yourself with calcium, you'll slowly wear away at those bones because you need the calcium to use in other parts of the body. So here is what is happening. What we're going to do is let's orient ourselves first. This is the sarcolemma, which otherwise known as the membrane of the muscle cell. And inside the sarcolemma, there is this yellow part that is going to be the sarcoplasmic reticulum. And then there are these other parts. We mentioned these before, these things called T-tubules. Think of T-tubules as basically folding in the membrane. And so the membrane actually comes into the inside of the cell. And what we're going to do is actually release calcium into the central area where the actin and myosin fibers are. And that is actually going to cause the muscle cell to actually contract. So calcium stored in these sarcoplasmic reticulum here, and it will stay stored in there until we get that message from the nerve, the neuron. The motor neuron is going to fire. It's going to get all the way to the end of the neuron. It's going to get to the neuromuscular junction. It's going to jump that gap, and it's going to actually then move across the membrane of the muscle cell or the sarcolemma. And when it reaches there, it's going to actually enter the cell through these T-tubules, and that is going to trigger the calcium that is stored in these nice little pockets here to all of a sudden be released, and it's going to float out, and it's actually going to interact with the actin and myosin, allowing them to actually contract. So again, yeah. What is actually happening here is it binds to a molecule known as troponin, and you're going to see this. So we're zooming in here. Troponin is this little red molecule here, okay, and it's an accessory protein on top of actin, so obviously much smaller than the actin fibers, but this little molecule here. And it actually has a binding site for calcium. So as you can see here, it changes shape. So this is its normal shape, and then when it binds to calcium, it actually changes shape. So that's significant, that binding to calcium and it's changing shape, because we also have another molecule known as tropomyosin, and that's that long, skinny molecule that wraps around. And what the tropomyosin does is it blocks these things called binding sites, those little yellow dots there. Those binding sites are the places where the myosin can actually, the myosin heads actually want to bind to the actin, and they actually want to interact, forming what's called a cross bridge. And right now, the myosin cannot form a cross bridge because this tropomyosin, this little thin fiber, is actually covering the binding sites. But we're going to see what happens, that when we actually bind this, what we're going to do is we're actually going to make the troponin molecule change shape, which is then going to move the tropomyosin. So when all the troponin have these calcium bound to it, we're going to move the tropomyosin, and what you can see here is now these little yellow binding sites are now open. And when they're open, we now can form these cross bridges between the myosin and the actin molecules. And the rest will happen automatically. As soon as you form these cross bridges, the ATP will start actually moving these myosin heads, and it will start walking along this actin. And as long as calcium is in the muscle cell, it's released there, that muscle will continue to contract and contract and contract. If you want the muscle to stop contracting, you have to quickly reabsorb the calcium, which takes no time at all, because again, this happens very, very quickly. So one aside that I actually think is an interesting aside is that you may have heard of something called rigor mortis. Rigor mortis is something that happens to you after you die, or it happens to anybody after they die. It happens to any mammal, actually. It doesn't have to be just humans. And essentially what happens is that over the course of 12 to 24 hours after you die, your muscles become incredibly stiff. And then eventually you lose that. It does dissipate after a while, but your body becomes very stiff. And you've probably even heard of, people call a dead body a stiff because it's of rigor mortis. What's happening there is that the sarcoplasmic reticulum that stores the calcium does a really good job of storing calcium, but it needs to actually actively take up calcium all the time. It's got pumps on it. And eventually, after you die, the calcium slowly starts to leak out into the muscles. And so the calcium will actually bind to the troponin inside your muscle cells. It will move the tropomyosin, and you'll actually form these binding sites. And you may say, well, why doesn't the muscles move? Well, at the point that this happens, there's no more ATP. You've run out of energy because the cells are dying, dead. There's no more oxygen. So you will form these binding sites between the actin and myosin, but you will not be able to actually move the head. So the muscles don't actually contract, they just become super rigid. They cannot be stretched or pulled or anything like that. Then over the course of a little while longer, those bonds start to break down, and actually the proteins start to break down because just general degradation of the proteins. So that's just an interesting aside, not necessarily a test question, but it explains why that happens. And again, we'll talk a little bit more closely about how this whole mechanism takes place. But the key, again, is that calcium is released from the sarcoplasmic reticulum. It binds to the troponin molecules. The troponin changes shape and then moves the tropomyosin. And then once the tropomyosin is moved, you can form the cross bridges, and the cross bridges will actually start the contraction. All right, thanks, guys. Bye.